Hello guys and girls, this is a final exam review for Human Development and Aging. The final that you guys will do on Monday is going to be very similar to the midterm. 8 questions, short answers, similar point value. The material that it will cover will be from the end of the pregnancy section up until the brain aging lecture that I posted for you late last week. We didn't cover the obesity or exercise subsections in pregnancy, so those are eligible for the final exam as well. Now we'll jump into content because there's a ton of stuff to talk about. This is not going to cover everything. Please make sure to review your notes and let me know if you have any questions. The first main idea that we talked about in the aging section was the value of life. You guys have it as the cost-benefit versus age. That's written on your slides, but a more apt description is value of life. The value of life theories were ways that we tried to understand why we aged. They don't propose mechanisms, but we do get into progressive detail as the course goes on. So these are starting to illustrate why we age. We're starting to think of the scenarios where it would be a benefit or a detriment to be alive. The first scenario says that there's always an advantage to being alive. You have more offspring, you can nurture those offspring, and this is essentially what was proposed by Darwin. As I go through these, start thinking about some of the evidence for and against these. I'll address that in a second, but it's good to think about that as we talk about these theories. The second theory encompasses scenario 2 and 3 on your page, but I've renamed them 2A and 2B to avoid confusion with the blueprints that we talk about in a second. And also because these two scenarios are very similar. The differences are minimal. They say that there's either no or very slight benefits to being alive past a certain age. So once an organism reaches reproductive maturity, that's clearly a benefit. The importance of that organism being alive starts to decline. The third scenario suggests that there is an active detriment to a long lifespan. Instead, there is an optimum lifespan of some kind, and processes in the body regulate lifespan to try to achieve optimum lifespan. Now, there is some evidence for and against each of these theories. We really came to believe that scenario 3 was the most important because this idea of a program seems to be a common one that we've encountered as we went through the material. The blueprints were an attempt to put a framework to these theories, try to figure out ways that the body would act if this were the case, and then places that we could intervene or observe to verify the theories. So blueprints 1 and 2 were the most basic. They are not program related. They're called error theories, and they typically relate to scenarios 1 and 2. So blueprint 1 was that you just accumulated damage over time, and then eventually you accumulated too much damage, you aged, and you died. The problem with that theory is that if it were just accumulating damage, many organisms should do that at the same or similar rates, and therefore have similar lifespans. But the example that we used of the rat versus the squirrel illustrates nicely that relatively similar organisms don't have similar lifespans. So we're not entirely convinced that it's just damage accumulating. We think that there is a gradual decline and then death. Error theory number 2, or blueprint number 2, included this idea of a repair mechanism, saying that the difference in lifespan could be accounted for by the difference in effectiveness of the repair mechanisms to counteract this damage. And so that jives with what we know about gradual decline and then eventually death. What really sticks out are blueprints 3 and 4, which hint at the idea of a program. we are starting to believe that this is more the case with aging, that it's not just individual tissues that age or individual things that contribute to aging, but that for some reason the body decides to limit its lifespan. It's written into our genetic code. And so this program will try to limit lifespan because it becomes a detriment at some point later on. And it could be that you're consuming resources that your offspring would normally use, or maybe it's to refresh the gene pool. The rationale for it remains unknown, but we get insight from a lot of these experiments that manipulate only one gene and then observe changes throughout the body in classic aging indicators. That seems to point to a program. The biggest point to a program would be calorie restriction, and we'll talk about that in a second. So we first started looking at these things individually, these theories of aging. We looked first at Ross, which is thought to be the main basis for the scenario 1, or the general wear and tear theory. The higher your metabolic rate, the more Ross you would produce, the more damage you would accumulate, and therefore the shorter your lifespan would be. We did see that when we increased Ross, lifespan decreased, but what was controversial is that when we got rid of Ross altogether, we didn't observe an increase in lifespan. So something was still limiting the lifespan. Ross didn't tell us the whole story. We thought that it could be hormonal, this idea that aging is maybe regulated or starts to occur when sex hormones decrease with age. And indeed, we looked at growth hormone and testosterone, and they decrease with age. But when we give either of those hormones, it seems to shorten lifespan. So no conclusive story to tell there, but we do get some important information from the Cynthia Kenyon TED Talk about how hormones can positively influence lifespan. She did the work that looked at IGF-1 signaling and lifespan. So if we remove the receptor for IGF-1, we see an increase in lifespan, saying that this could be one thing that is part of the aging program. If you remember way back, we also talked about target of rapamycin. We know this mostly from the protein synthesis research from last semester, and we haven't been able to include it into our main overall program, but it does seem that activating TOR will increase longevity for some reason. It's a really interesting area for further study, and we don't know exactly how this fits in, but part of the program could involve TOR. And so we're left with all these individual changes with aging, and we have to wonder if they're related. Are all these things that change independent, or do they all originate from some overseeing mechanism? And we start to get an idea that it's probably a program because these individual mutations in our genetic code cause widespread changes in the body that you don't think would happen without a program. So just modifying one gene, either the DAF2 example or progeria and Werner syndrome, caused changes that are indicative of aging all across the body in really young individuals. So changing that gene really changed and accelerated the program for some reason. Now, none of these individual mechanisms of aging really addressed the physical limitations. And so we also had to acknowledge that there seems to be a limit to the time or the number of times a cell can divide. And this is called the Hayflick limit. He found that cells really only divide a certain number of times before they grow old and die. And it was thought back in the 60s that this was due to contamination in the cell culture dishes, because the dogma, so to speak, was that cells could live forever if you gave them enough nutrients and supplies. So the experiment that Hayflick developed took two sets of cells, male versus female, so you can readily identify these different cells. He grew them essentially to different ages, so that the female cells develop or multiply 10 times, and the male cells multiply 40 times. Froze them at that certain age, and then recombined them in the same media to see if there was contamination in the media, would it kill all the cells? That's what you would think, right? Now at the same time, he put control cells in the same media but in different dishes to look to see if there were any effects there as well. And the only cells that seemed to die were the older male cells. Somehow they seemed to remember how old they were, as though they progressed partway through a program and then picked up right where they let off. There's no reason to think that contamination would kill only the male cells. It points to there being some inherent limitation. And we think that the reason underpinning this now are telomeres. So you can see telomeres illustrated here. They're the green end caps on this chromosome, and they're required for cell division. So you need to be able to attach to the telomeres to pull the chromosomes apart and then replicate them every time the cell divides. But what we see is that with each cycle, these end caps get smaller. Eventually they reach a point where they can't allow cell division to occur. And so at that point the cell becomes senescent, and we typically observe senescent cells in older people, so we think that that contributes to aging. Now we do normally see that older cells have shorter telomere length, and on the flip side we see that long-lived humans, typically centenarians, seem to have longer telomeres. So this might be a heritable trait that establishes your baseline longevity. We really switched away from these individual mechanisms of aging into a program model when we looked at calorie restriction. It's really the ultimate model that points to blueprint number 4 as being correct. The regulated program of aging, because calorie restriction is something outside of the cells of our body that influences the function inside our body. And not just the function, it influences lifespan and health span. And you can see that illustrated here with these monkeys, where the calorie restricted monkey looks far healthier, thicker coat, better posture, than the older monkey that eats a normal diet. You can see that this resulted in an increased survivability with calorie restriction. More monkeys lived longer. But not only that, they seemed to have better health as well. The calorie-restricted monkeys had fewer incidences of disease, and at any given point in time, there were more animals with no disease in the calorie restriction group. So it seems like a really weird idea that limiting energy intake would ever affect health span and lifespan positively. We rationalized it by thinking of having offspring in a famine situation. It's probably a bad time to reproduce because you might not be able to feed all your offspring, and you wouldn't have what we call very good genetic success, or rather reproductive success. So if there was a way that the body could respond to this situation and delay reproduction until a time when food was more available, there would be more reproductive success and a better chance of passing on your genes. So it makes sense that a program to alter reproduction would exist, and maybe it's a side effect of this program that lifespan is also increased. So we wanted to know why this happens. What are the mechanisms behind calorie restriction? And we identified sirtuins as key players in this response. So we know that in yeast, if you calorie restrict, a yeast will live longer. But if you calorie restrict without the sirtuin gene, nothing happens. Those yeast will live just as long as normal wild-type yeast eating a lot of glucose. And so what is sirtuin? There are a lot of them. In mammals there are 7 different kinds, but what's important is that they respond to NAD in the cell. Now if you remember back to metabolism, NAD is the shuttle which carries electrons into the electron transport chain. It's like a boat that ferries people across a river or across an ocean. So when food is available, there's electrons available, and when there's electrons available, NAD will carry them. It'll be in the form NADH. When there's little food available, then there's fewer electrons, and so more of the NAD will be in the NAD form. This is important because it activates this SIRT1 protein and is the main reason that we think calorie restriction works. Now, it doesn't completely discount that insulin and IGF-1 might also mediate some of this calorie restriction response, but we'll talk about that in a second. So we know in setting up this program from the Cynthia Kenyon TED Talk that normally the DAF2 gene, that's the IGF-1 receptor, it normally inhibits FOXO activity. And if you reduce DAF2, either by knocking it out or reducing IGF-1, then you remove that inhibition on FOXO, and then FOXO can do its job of activating longevity genes. It can cause organisms to live longer. Now it seems that calorie restriction does something similar. It seems to activate NAD because we continue to metabolize food even though there's a lower food supply. This increased NAD will activate the sirtuins, and then sirtuin activates FOXO directly by deacetylating it. Now, deacetylating is just a way to indicate to the cell what activity state a protein has. It's a tag, just like phosphorylation is a tag. So SIRT1 gets rid of the acetyl group, and in this case, when FOXO does not have the acetyl group, it's active, and then it can activate those longevity genes. Now, insulin and IGF-1 are not out of the picture because it's possible, at least it's certain, we know that calorie restriction reduces insulin in the blood. It also reduces IGF-1, and so it's possible that some of the influence on longevity goes through this left-hand arm of the pathway. Some of it might work through removing the inhibition of DAF2 on FOXO. What's neat about this, though, is that these proteins are in every cell in the body. They're part of our genetic code, and so it's more or less a similar aging process in all cells. So we really get the idea that it's not just treating vision or treating brain function or treating muscle. If you can treat the program, then you have a much higher likelihood of success in all of those downstream tissues. Now, what does FOXO do? We say it increases longevity, but what does it do? We know at the whole body organism level that it increases lifespan, but at the cell level, we're still not quite sure, although we see that it seems to pause cell division and reduce cell death. So you can see that whenever FOXO and SIRT1 are both active, more cells are paused in their division cycle, and fewer cells undergo apoptosis, which is cell death. Now we got a great lecture on Resveratrol, which is our first attempt at mimicking calorie restriction. The problem with calorie restriction is that you have to calorie restrict, and no one wants to do that. It's not a pleasant feeling to have to not eat enough food every day. So Resveratrol is a way that we think we could stimulate all the benefits of calorie restriction without having to do the calorie restriction. Now we see that adding Resveratrol to yeast increases their lifespan, and we're fairly certain that it works through SIRT1, I've summarized the three main points here, but I encourage you guys to go look at the actual data in Marie Soleil's lecture. We think it works through SIRT1 because we see when we supplement with resveratrol, there's more SIRT1 in the muscle. And we even see this in humans. We know this works. If we get rid of the gene that makes the sirtuins, then resveratrol has no effect on longevity. So we took out that critical link in the chain, and it couldn't send the signal to increase longevity. Finally, the third point is that resveratrol and calorie restriction aren't additive at all. Calorie restriction has already activated this response as much as it will be activated, and resveratrol can't do anything more. So if we go back to our summary, we would add resveratrol right here. We think that it increases SIRT1 protein. That allows it to deacetylate more FOXO and possibly increase longevity. The unfortunate thing is that it's not feasible in humans. You have to drink way too much red wine in order to get enough resveratrol to match the levels that are used in these exploratory studies. Thousands of liters of wine a day would probably provide more of a detriment than any benefit from the resveratrol that you take in. Not only that, but resveratrol seems to be broken down into less active metabolites in humans. The liver breaks resveratrol down into these other forms that don't do as much of the tissue, and even though it's a positive relationship, as you increase the dose, you will increase active resveratrol. When you increase above 5 grams, the negative side effects become much more noticeable, and the downsides, I would think, outweigh the benefits of supplementation in this case. So we're looking for new compounds that might not be broken down or might be more potent, but the search is on, and we're not convinced that resveratrol will work itself in people. We pulled back a bit and started talking about the tissues again. We've realized that there's a general decline in function in all the tissues of the body, and we're starting to see that they might be related to the activation of this program. We really touched on muscle, as well as some other tissues, but sarcopenia is something that a lot of older people struggle with. That's general frailty and weakness in the muscle. And so whether or not we know what the aging program does, we wanted to see if there was a way that we could fix it or help these people. And certainly it seems that training does that. It reverses or partially addresses sarcopenia. So we see increased VO2 max, increased muscle size, and increased muscle strength. We don't know why that happens. This study said that it increased phosphorylated FOXO, and we have no idea what that means. We've only talked about acetylated FOXO. And it also says that it decreases PGC1. Again, that goes against everything we know about aerobic training. PGC1 is the overseer of the aerobic response, so we're not sure why it would ever go down. But the point is that it seems to have benefits at a whole body level. The same thing can be said with resistance training. We get an increase in muscle size, an increase in muscle strength, and hopefully that can translate to mobility and quality of life. Now, it's a little bit odd, though, that these things work, because we've noticed that elderly muscle is less responsive to exercise. They have an inherently lower protein synthesis, and that's not stimulated by exercise. So we took a look at supplementation as well. We often think of protein supplementation along with resistance exercise. And while exercise increased muscle size, adding protein didn't give you any additional benefit, which again is weird. It goes against a lot of what we know about protein and resistance exercise. What was really cool, though, in this situation was that we seem to get a benefit from protein ingestion after we've supplemented with omega-3s. So for some reason, supplementing with omega-3s increases the ability of protein to stimulate muscle protein synthesis. If we supplement with just a normal, boring corn oil, we get the same response before and after supplementation. But if we supplement with omega-3 fatty acids, you can see the response is much higher after the supplementation protocol. So after 12 weeks of supplementation, for some reason, omega-3s made the muscle more responsive to amino acids. So this seems to be an important point, at least for elderly people, to consider when thinking about how to help them increase their mobility and deal with sarcopenia. Omega-3s really don't stop there. They also seem to help with Alzheimer's. So in mice that are meant to develop Alzheimer's, that overexpress the beta amyloid proteins that cause beta amyloid plaques to develop, we're pretty certain that the appearance of those plaques relates to Alzheimer's disease. When these mice were given either no DHA or lots of DHA, we saw a decrease in the amount of that bad beta amyloid protein that accumulated in the brain. Remember, the longer proteins are harder to get rid of. They seem to form more plaques. Not only did we see a decrease in the long beta amyloid protein, but we also saw a decrease in plaque formation. So clear benefits of supplementing with DHA, at least in these mice. The other thing to consider is that having no DHA, absolutely none, doesn't provide a benefit, and in fact it could be a detriment to Alzheimer's in these mice. It increases the normal, quote-unquote, form of beta amyloid protein. We don't know what that does, but it's not something that you would want to have happen. Another really interesting way to deal with Alzheimer's disease, and I'm not going to go through all of the brain aging stuff because we just talked about it in the last video, but it's really cool that we can now bring calorie restriction into this picture as well. It seems to have widespread effects throughout the body, improving health span and improving lifespan, and it also seems to improve Alzheimer's in a different mouse model that's meant to get Alzheimer's as well. Now, this specific study compared calorie restriction to intermittent fasting, and it found that calorie restriction seems to be really good at lowering the levels of those beta amyloid proteins. Now, these are markers, and we're concerned about them because having too much of these mean that we're going to develop plaques, and we think that plaque formation is related to the development of Alzheimer's disease. So here we would definitely pick calorie restriction as our main therapy, certainly not intermittent fasting because it doesn't do anything. Or does it? If we look at the whole body changes, sure, we don't see changes in the beta amyloid protein, but both the time to complete the maze and the path that these mice took to complete the maze were all improved regardless of which calorie restriction protocol you used. Now, it's debatable on whether or not this is cognitive function, but certainly mobility and problem solving come into play, and these are things that not only these mice but all older people would want to improve. And so it has to change our perception of Alzheimer's a little bit. We've thought universally that those beta amyloid plaques cause Alzheimer's, but here we're looking at a way to interrupt the negative effects of those beta amyloid plaques. This suggests that intermittent fasting protects against those intermittent plaques. So a slight tweak to our understanding of Alzheimer's disease. Now I'm going to cut the review off there. I don't want this to be too long for you guys. This certainly didn't cover everything, and I hope that if you have questions you will email me. If you don't, and I don't hear from you, then best of luck studying, and I will see you guys on Monday.